Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so we just got to the point where the B cell had found its antigen. Okay, so the antigen had percolated through the B cell follicles. It had searched and searched for a B cell that had the complementary B cell receptor for this antigen, and then when it found it, it bound to this B cell receptor. And then what that triggers is the B cell endocytoses the whole B cell receptor with the antigen bound to it and breaks down that antigen and presents a fragment of that antigen on the major histocompatibility complex class 2 here. Okay, now B cells are a professional antigen presenting cell. So what's going to happen is they're also going to present co-stimulatory molecules. So in addition to the antigen coming in the lymph that is approaching the lymph nodes, you're also going to get all sorts of pathogen-associated molecular patterns, PAMPs, coming, which will activate pattern recognition receptors within the B cell, and this will cause the B cell to express the co-stimulatory molecules. What's then going to happen is our B cell here is going to interact with this T helper 2 cell, okay, in the same way as usual. So here comes the MHC class 2 now on the B cell, and it's got this antigen fragment that this T helper 2 cell has a T cell receptor which is complementary to. So this is the same antigen all the way through that we've been talking about basically, and the B cell receptor binds to that antigen, and also the T helper 2 cell has a T cell receptor which binds to this fragment of this antigen. Okay, so here is our T helper 2 cell here. Okay, right, so this is the T helper 2 cell. And then let's highlight the different bits of this complex in here. So here is the MHC class 2 in purple here. Okay, and then you've got this antigen fragment here, and then that's bound to the T cell receptor on the surface of this T helper 2 cell here. And then of course the T helper 2 cell also has a molecule of CD4 on its surface, which will also bind to the MHC class 2, which is here in purple. So this is MHC class 2. This molecule here on the surface of the T helper 2 cell is CD4, and it's binding to the major histocompatibility complex class 2. Okay, so that's in green. And then what's also going to happen is the B cell here, which is still inactive at the moment, is going to also have presented co-stimulatory molecules on its surface. So let's say this is either B7.1 or B7.2, and this is CD40 here. So this is CD40, and then it's going to bind to receptors on the surface of the T helper 2 cell. Okay, so again this is CD40 ligand, which binds to CD40, and this is CD28, which binds to either B7.1 or B7.2, which is on the surface of the B cell, and has been expressed there since the B cell had its pattern recognition receptors activated by the presence of the PAMPs. Okay, right, so let's highlight these up. So we'll have B7.1/7.2 in orange, we'll have the CD28 in blue, we'll have, I might try using yellow just once. So here's CD40 in yellow. Has that shown up? Oh, it just about has actually. And then we'll have CD40 ligand in turquoise here. Right, okay, so what does this lead to? Well, basically, this is going to lead to the activation of the B cell. Okay, so let me run through what's happened again. So the B cell was sitting quietly in its B cell follicle when this antigen arrived, and this antigen just happened to be complementary to its B cell receptor. So the antigen binds to its B cell receptor, it's internalized, the antigen is broken down, and a fragment of it, well, many fragments of it will be presented on MHC class 2. Now, one of these fragments just happens to be the same fragment that you initiated the whole T cell chain reaction for. So we now have these T helper 2 cells, which have been made because we have an extracellular pathogen. So remember, we had the differentiation into T helper 2 that seems to happen when you have an extracellular or you're in need of a humoral immune response, and we're not clear really on how this differentiation actually occurs, although what we do know is that interleukin 4 appears to play a very important role in helping the differentiation of T helper 0 cells into T helper 2 cells. Okay, but we're not clear on where the interleukin 4 comes from, or even how that signifies that this is an extracellular pathogen rather than an intracellular pathogen. Okay, so then what happens is the B cell, which now has this antigen fragment mounted on its MHC class 2, binds to the T cell receptor on the surface of the T helper 2 cells, and then obviously CD4 also binds. Then the B cell has also expressed co-stimulatory molecules, B7.1 or B7.2 and CD40, because it's a professional antigen presenting cell and has many pattern recognition receptors which are being activated by pathogen associated molecular patterns which are coming to the lymph nodes via the lymph vessels. Okay, and these bind to their complementary receptors on the surface of T helper 2. What this then triggers is it causes the activation of the B cell, and this B cell now divides and divides and divides into many different B cells. So you're going to get multiple B cells here. So here's 1, here's 2, and again I'll just draw 3. Now, you might think, oh brilliant, this looks a nice simple process, but here's where something really clever happens, and this is complicated to understand. This is probably the most difficult concept of what we're going to cover in this video. Okay, so we are now going to go through a process known as somatic hypermutation. Okay, so what happens is when these B cells divide, so when this B cell divides to make a whole population of B cells, what am I doing here? Hyperpolarization is what I'm trying to write. Hypermutation. Okay, right, so somatic hypermutation is what's going to happen next. So basically, when you produce this population of B cells, they're not all going to end up identical. Instead, you're going to introduce a whole bunch of little random mutations into the B cell receptor, into the antigen binding domain of this B cell receptor. Okay, so all of them are going to end up with slightly different B cell receptors. They're almost identical, but they're not quite identical. Now, what's going to happen? Well, basically, if we now think about what's happening in the B cell follicle, and this is important. So if this is the B cell follicle within the lymph node, so I've taken a single B cell follicle out here, then what you're going to get is a whole population of these B cells. So we started off with a single B cell within this B cell follicle, so I'll label this up. Okay, B cell follicle. Okay, so we started off with a single cell. Now it's going to divide and divide and divide and divide. So you're going to end up with a whole population of these cells. Okay, and these appear slightly different to the resting B cells, basically. So you can see this collection of new cells that are all made from a single B cell that have been activated by this process. And when you look at it down a microscope slide, and this is what's known as the germinal center of the B cell follicle. Okay, so this is called a germinal center. So a germinal center is this collection of B cells which have all been formed by the division of a single B cell which has been activated because it's actually found its antigen, basically. Okay, now all the cells in this germinal center will not have identical B cell receptors. Instead, they'll all have slightly different B cell receptors. Now, there is another cell in B cell follicles that is going to become important now. So this is what's known as a follicular dendritic cell, and again it's got absolutely nothing to do with neurons. It's just a cell which has dendrites. Okay, so this is what's known as a follicular dendritic cells. Follicular dendritic cell. Now, what do follicular dendritic cells do? Well, basically, they pick up antigen and present it all over themselves, okay? So this is a follicular dendritic cell. So, as antigen percolates through the B-cell follicles, the follicular dendritic cells are going to catch the antigen and hold it on their cell surface membranes. So let's say this is the antigen here, and it's basically all over these follicular dendritic cells. Now, why is this useful? Well, basically, this means that there is a large supply of this antigen for which these B cells have complementary receptors for. Okay, and now what's going to happen is that this huge great population of B cells in the germinal center, which all have slightly different B cell receptors, are going to compete for the antigen that is on the surface of the follicular dendritic cells. Okay, so we're going to start a process of natural selection here. So they all have separate B cell receptors on their surface, which combine to this antigen here, but there is a limited amount of antigen. So which of them are going to get the antigen? Well, it's going to be those that had mutations in their B cell receptor, which meant that the affinity of the B cell receptor for the antigen was actually increased. Those which had mutations in their B cell receptor which resulted in lower affinity or even maybe a loss of binding altogether, those are not going to get the antigen because it simply is not going to bind. So some of the B cells in this population within the germinal center will have undergone mutations in this process of somatic hypermutation that meant that their B cell receptors, shown here, so this is a BCR for short, actually have a higher affinity for the antigen now, and they will take up loads of this antigen. Okay? Whereas those that don't, those that underwent mutations that caused a reduction in the affinity, those won't get the antigen, basically. Okay, now, what's going to happen to these ones that do undergo, which do have mutations which increased the affinity of their B cell receptor for the antigen? Well, basically, they're going to go through this exact same process again. They're going to internalize the B cell receptor with the antigen bound to it. They're going to break down the antigen. They're going to present it on MHC class 2, and they're going to go up to T helper 2 cells, which are also in the lymph node, and they're going to be activated. So this one will then be activated, and it will divide again. So this lucky old B cell is going to get T cell help, as it's called, and it's going to divide and make a whole bunch of B cells which are all based on it. So all of these will then also have B cell receptors which undergo somatic hypermutation. So the process occurs again. So what's going to happen is you're going to start with this B cell receptor, which remember was the improved one, and then you're going to make a whole bunch of mutations in it. And the idea is that now what will gradually happen is in this new population you'll end up with some B cells which have a B cell receptor that is even better than this one. Okay, so here comes this next one here, and let's say this one is lucky enough to undergo a mutation that makes it even higher affinity than this one. So it will lavish up antigen and it will go through the process again. Okay, right, so basically the B cells which get antigen and then interact with the T cells, they remain alive. In fact, they remain more than alive. They're given the signal to divide and divide and divide and produce a whole colony of cells which are based on them. Of course, they won't be identical to them because all the cells undergo somatic hypermutation when they divide, so that all of the cells are slightly different, but all of them are based on it. Whereas the ones which undergo mutations that mean that their B cell receptor is less good at binding to the antigen, those will not get antigen, they will not present it to T helper 2 cells, and they will undergo apoptosis. So if they don't get T cell help, as it's called, so this process here is known as T cell help. If they don't get T cell help, they die, basically. So only the ones which are best at picking up the antigen are going to survive. And gradually what you're going to do is you're going to select, therefore, for B cells which have B cell receptors which have very high affinity for the antigen, okay? And gradually you're just going to get B cells which have a higher and higher affinity, because even if we have a B cell that has a good affinity, some of these will have absolutely great affinities, and there's only a limited amount of antigen. So the antigen will end up bound to the ones with the great affinity rather than the good affinity. So the good ones won't get it, and therefore they'll die. So you're gradually going to select for a B cell which has a fantastically good B cell receptor for binding to this antigen. And this concept is known as affinity maturation. Okay, now it is not fully understood what stops this cycle from continuing forever, but something stops it from continuing forever, and eventually one of these B cells that has an absolutely fantastically good B cell receptor is going to start dividing and dividing and dividing, and its offspring, instead of just going back through this cycle again, they are going to instead differentiate into plasma cells. Okay, well, they're going to differentiate into two types of cells. One is plasma cells and the other is memory cells. Okay, so we'll discuss plasma cells first and we'll discuss memory cells later. So, plasma cells then. And plasma cells have a very characteristic histological appearance. They have a nucleus that's very large but shoved into one side, basically. And all of this space here is then free for making antibody, okay? And then we'll have a memory B cell here, okay? And it just has a very large nucleus. All lymphocytes have very large nuclei. Okay, so this is a memory B cell. Okay, so let's discuss what plasma cells are going to do then. Okay, but we'll do that in the next video.